My dear students, we are going to discuss today a important chapter from protein metabolism, that is protein digestion and absorption. Proteins are organic compounds formed of amino acids joined together by peptide bonds. In contrast to carbohydrate and lipids, whose major function is to provide energy. The primary role of amino acid is to serve as a building block for the synthesis of body proteins. Proteins are too large to be absorbed by the intestine. Therefore, they must be hydrolyzed at first to give amino acids which can be easily absorbed. Among the importance of the protein or functions of protein, we can list few, that is synthesis of tissue protein, example contractile proteins of muscles, collagen, etc. Second, synthesis of cell organelle as cell membrane, receptors, etc. Third, synthesis of enzymes. Fourth, synthesis of milk protein. Fifth, synthesis of protein hormones. Sixth, synthesis of plasma proteins. And seventh, synthesis of nucleoprotein. Now we will go towards the digestion process of protein. There are three organs which produce the enzymes needed for protein digestion. Those three organs are stomach, pancreas, and small intestine. The dietary proteins which we consume as food, they are masticated, chewed, and swallowed through the buccal cavity. There are no enzymes practically in the buccal cavity for digestion of protein. We have enzyme for carbohydrate digestion like amylase and lipid digestion like lipase. But protein digestion it starts right in the stomach. The enzymes which are present in the stomach which take the start of the digestion of protein are pepsin and renin. Pepsin enzyme which has got maximum activity, that is its optimum pH is between 1 to 2. It is secreted by the body chief cells, serous cells of the stomach as inactive proenzyme, also we call it as zymogen. That name of the zymogen is pepsinogen. Pepsinogen is activated to pepsin at first by gastric HCl, then by autocatalysis by the pepsin molecule with that have already been activated. This is called as autocatalysis. Pepsin is endopeptidase, that is, it acts on the amino acids in the middle of polypeptide chain. It acts on those peptide bonds of amino acids which are in the middle of the polypeptide. It hydrolyzes the bonds formed by aromatic amino acids. For example, tyrosine, the bond peptide bond formed from the carboxylic group of tyrosine with its adjacent amino acid is hydrolyzed by pepsin. Pepsin acts on pH 4. The optimum pH is 4. Renin the second enzyme in the gastric juice which acts on the protein is renin. That optimum pH for renin is again acidic, it is about 4. Renin causes coagulation of milk proteins. Renin acts on casein, which is the main milk protein. In presence of calcium ions, renin converts casein into insoluble calcium caseinate, that we call it as milk clot. The digestion of calcium casein is completed by pepsin. That is, pepsin will act on calcium caseinate. Renin is absent in adults and present only in infant stomach. Renin is important for infant because the formation of milk clot prevents the rapid passage of the milk from the stomach. This gives the baby the sense of fullness. Second organ which is responsible for further digestion of the protein which comes out of stomach is intestine. In this intestine, there are enzymes from two organs, pancreas and intestine, which act on protein in the intestine. Pancreatic enzymes, these include trypsin, chymotrypsin, carboxy peptidase, and elastase. In the old literature, gelatinase and collagenase were also discussed as the intestinal enzymes in mammals, but these are actually the enzymes which are because of bacteria. Their origin is in bacteria. They are not directly concerned with mammalian protein metabolism. So the intestine, as we said, there are four enzymes which act. Those are tyrosine, chymotrypsin, elastase, and carboxy peptidase. Trypsin, its specificity is it will hydrolyze the peptide bond formed by the carboxylic group of the basic amino acids arginine, lysine, whereas chymotrypsin is having affinity for the peptide bond formed by the carboxylic group of tryptophan, tyrosine, phenylalanine, methionine, and leucine. It hydrolyzes the peptide bond formed by these amino acids. Elastase, although its name gives some affection for the elastin, but it acts on the amino acids like alanine, glycine, and serine. All small molecular weight amino acids are attracted by this enzyme. The carboxy peptidase is an exopeptidase. All these three, what we have discussed, they are endopeptidases. Endopeptidases are those enzymes which act on the peptide bond of a polypeptide in mid, not on the everything. Whereas exopeptidases are those which act on the peptide bond contributed by the extreme amino acids, that is N-terminal amino acid or C-terminal amino acid. Here our fourth enzyme is carboxy peptidase. It is specific for it will be hydrolyzing the amino acid present on the C-terminal, and it is having an affection for the amino acid like alanine, isoleucine, leucine, and valine. Now we will take up the endopeptidase one by one. The trypsin is the endopeptidase. Its optimum pH is alkaline. When the chyme from the stomach enters into the small intestine, that is duodenum and ileum, there the pH is alkaline because of the carbonate secreted by the pancreatic juice. So the pH is 8, and the trypsin which is secreted in the pancreatic juice is in inactive form, in zymogen form. The name of that proenzyme or zymogen is trypsinogen. Trypsinogen is activated to trypsin by two systems, two methods. At first, by entropeptidase enzyme produced by the intestinal mucosa, then autocatalytically by the trypsin molecules which are already been activated by entropeptidase enzyme. Trypsin is a endopeptidase, hydrolyzing the peptide bond formed by basic amino acids, for example, lysine and arginine. It releases smaller peptides and more free amino acids. Trypsin also acts as activator for all other inactive zymogens produced in the pancreas, present in the pancreatic juice. Next is chymotrypsin. Chymotrypsin is secreted as an inactive proenzyme, that is inactive zymogen, namely chymotrypsinogen. It is activated by trypsin enzyme. It is an endopeptidase hydrolyzing the peptide bond formed by aromatic amino acids. Its action is similar to that of pepsin. Carboxy Peptidase is exopeptidase enzyme. It is secreted as an inactive proenzyme or zymogen, namely pro-carboxy peptidase, and is activated by trypsin, as we already told. It is an exopeptidase, acts on the periphery of the polypeptide chain. Carboxypeptidase hydrolyzes the peptide bonds adjacent to the free carboxylic group of the polypeptide. The polypeptide chain releasing each time a single free amino acid. The last enzyme from this group is elastase. It is secreted as an inactive proenzyme, pro-elastase, and is activated again by trypsin. It is an endopeptidase. It, in spite of its name, it hydrolyzes the peptide bonds formed by small amino acids, example alanine, glycine, and serine. Then we go for the next stage of digestion is absorption of these amino acids. The ultimate product we get from the action of this enzyme, that is pepsin, trypsin, chymotrypsin, elastase, and carboxypeptidase, we get a good amount of protein converted into mixture of amino acids. These amino acids, these are in the intestine. There are some other enzymes like aminopeptidase, dipeptidase, tripeptidase. These enzymes also act on the peptides, on their substrate, and ultimately convert maximum part of the protein into amino acids. These amino acids formed by the digestion of the protein is then subjected to absorption by the intestine. Absorption under normal conditions, the dietary proteins are almost completely digested into amino acids. These amino acids are then rapidly absorbed from small intestine into portal circulation. In newborn infants, proteins may be absorbed as such, that is, native protein can be being absorbed. Such proteins absorbed, native protein absorbed, where gamma globulin present in colostrums are absorbed, which give immunity to the baby. The passive immunity to the baby because of the mother's milk is because of this, that native proteins are being absorbed by the mucosa, intestinal mucosa. Colostrum is the milk secreted from mammary gland, and in the first few days after the labor, the milk form secreted is called as colostrum. Jejunum, and here then we go to the site of absorption. The site of amino acid absorption is jejunum and ileum, part of the small intestine. Now there are different mechanisms of absorption of the amino acids. The amino acids are absorbed either by active transport mechanism where energy is required. So all L type of amino acid present in the natural protein, they are absorbed by active transport. Or the other mechanism is by passive transport mechanism, which needs no energy, and D amino acids are being absorbed by this way. Now, active transport for L amino acid is almost same as that of absorption of active transport of glucose by spending energy. In active transport of protein, there are two systems acting. One is glutathione transport system or carrier protein transport system. Energy is required as derived for both the system from ATP. In case of active transport, we get energy from sodium pump. For each amino acid, one ATP molecule is utilized. Two mechanisms are involved: carrier protein transport system and glutathione transport system. Energy required is in active transport, the energy required is derived from the sodium pump. For amino acids in a small intestine by a mechanism similar to that of glucose absorption. That is, the carrier has one site for the amino acid and the another site for sodium. There are there are five types of carrier protein groups. Each transporter group of the closely related amino acids. The groups of amino acids are small neutral amino acid, large neutral amino acids, basic amino acids, acidic amino acids, and amino acid. This diagram shows that the active transport of amino acid is by ATP with the help of sodium potassium pump is very much similar to that of absorption active transport of glucose, where two potassium ions they enter and in place of three sodium ions, and the one ATP molecule is hydrolyzed to ADP plus ATP. Then the carrier protein binds to, it has got two sites, two binding sites, one for amino acid, other for sodium. So the sodium binds to the carrier protein, then amino acid binds to the same carrier protein, and then it is diffused. It goes inside the cell where it gives away its three sodium ions and amino acids. These sodium ions are, by their concentration gradients, are again sent out of the cell, and this way this system works that for each amino acid one ATP molecule is being utilized. The amino acids react in glutathione transport system. The first step is the amino acid reacts with the glutathione, forming gamma glutamyl cystinyl glycine at the surface of the cell to form dipeptide that is transported across the membrane into the cytoplasm of the cell. This reaction is catalyzed by a gamma glutamyl transferase enzyme, GGT enzyme. The subsequent reaction of the cycle serves to regenerate glutathione. 3 ATP molecules are utilized for this reaction. This diagram shows that the amino acid out of the membrane, it first reacts with gamma glutamyl reacts in the presence of gamma glutamyl transpeptidase to form cysteine glycine. This cysteine glycine is then hydrolyzed into glycine plus cysteine. The cysteine and glycine are then reutilized by the cycle. The amino acid which comes inside the cell as glutamyl amino acid is hydrolyzed. It gives amino acid to the cytoplasm, and other product formed from gamma glutamyl amino acid is oxoproline. This oxoproline, with the expenditure of an ATP, is converted into glutamate, and this glutamate, with again one more molecule of ATP, is converted into glutamyl cysteine, taking the cysteine from the cytosol. And gamma glutamyl cysteine then is converted into gamma glutamyl cysteine glycine with the expenditure of one more ATP molecule. This then get converted into two dipeptides. One is cysteine glycine and another is glutamyl glutamyl cysteine. Thereby, this cycle goes on. That is, cysteine glycine will combine with one amino acid, forming this tetra peptide that will split into two dipeptide, cysteine glycine and glutamyl amino acid. Cysteine glycine will give glycine plus cysteine. The glutamyl amino acid will be converted, is hydrolyzed into amino acid plus oxoproline. Oxoproline is converted into glutamate. Glutamate then reacts again with cysteine to form gamma glutamate cysteine. This way, this cycle goes on, and in each round, one amino acid is transferred from out of the cell membrane to inside the cytosol. This diagram shows the mechanism of this transport system, how glutathione is formed. First, glutamate reacts with the cysteine molecule, forming gamma glutamyl cysteine. One ATP molecule is utilized for this reaction. Gamma glutamyl cysteine then is converted into glutathione molecule by adding one more glutamate molecule, forming a glycine molecule, forming gamma glutamyl cysteine glycine, which is nothing but glutathione. So this glutathione formation is formed, requires two ATP molecules. Now, this is the reaction utilized for taking the amino acid from outside the cell to inside the cell. The other functions attributed to this glutathione transport system are: one is transportation of amino acid from the extracellular space into the cytoplasm. Second is synthesis of glutathione, which is an important reducing substance in the cells. And apart from this significance of this gamma glutathione transport system is diagnostic importance. That is, the enzyme GGT, gamma glutamyl transpeptidase, is found elevated in the blood under certain pathological conditions like cholestasis, immunoimpairment, or bile flow. Also, GGT is elevated in chronic alcoholism. Now we go to the hormones which contribute their role in absorption of amino acid from GIT. There are three main hormones listed in this series. One is gastrin, second is cholecystokinin, and third is secretin. Gastrin, its location is gastric antrum and duodenum. Major action of that gastrin is pepsinogen secretion and release of intrinsic factor from the gastric mucosa. The second hormone, cholecystokinin, CCK, its location is duodenum and jejunum. Its major action is pancreatic proenzyme secretion. The third hormone is secretin. Its location is duodenum and jejunum. The major action is pancreatic proenzyme secretion, pancreatic bicarbonate secretion. These are the three hormones which take part in the amino acid absorption phenomena. Now we go towards the clinical aspect of this amino acid absorption. The deficiency of the enzyme 5-oxoprolinase leads to oxoprolinuria. The allergy to certain food protein, that is, few persons have got allergy to milk, fish, or buffalo beef, is believed to result from absorption of partially digested protein. Defects in the intestinal amino acid transport system are seen in inborn error of metabolism. Partial gastrectomy, pancreatitis, carcinoma of pancreas, and cystic fibrosis may affect the digestion and absorption of protein. Protein losing entropathy, there is a excessive loss of protein through the gastrointestinal tract. Dipeptides and tripeptides can enter the brush border muscle cells.
Yeah.
Mention regarding food allergy, dipeptides and tripeptides can be enter to the brush border and muscles, mucosal cells, brush border mucosal cells. They are in they are immediately hydrolyzed into single amino acids. The transport they are transported into the portal vein. Rarely, larger molecules may pass paracellular between the epithelial cells and enter bloodstream. These are immunogenic, causing antibody reaction leading to food allergy. Few amino acid inborn errors we will mention here. When we study the individual amino acid metabolism, there we will be discussing this part of amino acid absorption in detail. We mention here are few diseases, inborn errors of the amino acid metabolism. One is Hartnup disease. Second is iminoglycinuria. Third is cystinuria. Fourth is lysuric protein intolerance and Ostau's syndrome. These diseases are given in detail by Vasudevan in his textbook of biochemistry. The references is given in chapter 18 and chapter 16. You can go for its clinical details of these enzyme errors. So this way we have touched almost all the aspect of amino acid absorption, amino acid, then proteolytic enzymes, the hormones which are concerned with amino acid absorption, and the mechanisms of amino acid absorption. So this will give you details of this. I request you to prepare a full note on this right now so that it will be helpful to you in preparing your university exams. Thank you.